That way I'm not going to forget. Okay. Well, good morning, everybody. I hope you had a great first day and figured out, got kind of taken with all the excitement of what we're doing and the wonderful projects that are going on and feel like you're getting a good start. Today, I'm really happy to hear, I'm going to be looking forward to hearing what you're going to do and your plan for the week. But before that, we always try to have inspiring talks. So we have an inspiring talk for you today from Dr. David Younger from A-Alphabio, and Todd is going to tell you a little bit about him before we start.
Yeah, thanks, Andy, and thank you, David, for joining us. And yeah, as a brief introduction, I met David, I think it was in 2017, just as you were getting this started, you were finishing graduate school and you were explaining how you're going to use this yeast-2 hybrid system to measure protein interactions. And I'm sure you'll cover this, but for those of you who don't know, yeasts are eukaryotes, right? And so they have sex because that's how we get rid of our rare alleles or recessives, those dreaded recessives that and yeast have two sexes and a cassette that kind of moves between them. So we'll hear more about that. But he was working on that in bioengineering, finishing up graduate school. First question I ask every graduate student founder is, are you going to get your thesis done? Because you're in this industry, you're going to be working with pharma and stuff, and having that PhD is actually an important degree. People, will listen to you, and they've been listening to David. And so from there, he got his PhD in bioengineering, did a really interesting one-year postdoc. University of Washington has a program for entrepreneurs where you can do a postdoc to get some funding so you can, pay your way to build your company. And so he used that time very well to cultivate a relationship with investors and started talking to big pharma. And since then, A-Alpha has to date raised about $51 million and has done some very significant deals with some major pharmaceutical companies in antibody development. So with that, David, why don't you tell us the rest of the story?
Great. Yeah, thank you so much, Todd. And thanks, Todd and Sandra and the rest of the team for inviting me and everyone for your attention today. Yeah, really excited to be here and tell everyone a little bit more about what we're building at A-Alpha. I want to start just by apologizing for my voice. I was telling folks before everyone left their breakout rooms, have a bit of a cold here. So I'll be kind of drinking some water and trying to keep from a coughing fit as we go. But please bear with me. Great. So just to kind of get things started, I want to give just a one slide, really quick overview of what we're building at A-Alpha, and then we'll go into much more depth. So at A-Alpha, we are the protein-protein interaction company. And we are really interested in all things protein-protein interaction, largely because protein-protein interactions are so important for just about all biological processes. So, for example, your cells recognize and attack viruses and other pathogens through the immune system, which is driven by proteins, by largely antibodies. Cells communicate with each other by secreting proteins and detecting those proteins on the surfaces of other cells. Protein interactions within cells govern a lot of how cells function and divide and grow. And because of this, the importance of understanding protein interactions and engineering protein interactions is really important, not only for understanding biology and understanding disease, but also for treating disease. And the two core approaches that we're taking towards protein interactions, the two mechanisms of action that we're pursuing at A-Alpha are broadly biologics and molecular glues. And we'll focus today on biologics, largely antibodies, but we're also interested in the discovery of small molecules that glue two proteins together that wouldn't otherwise interact with each other. So as Todd mentioned, we spun out from the University of Washington, from David Baker's lab at the Institute for Protein Design in 2018. To date, we've raised a little more than $50 million. We're based in Seattle. We have a team of just over 50 with broad experience both in the lab, so synthetic biology, protein engineering, and also computational experience. So data science, machine learning. Um, so with that, I really want to structure the talk in kind of three parts here. So the first is going to be the longest. So we're going to spend quite a bit of time on kind of going through what we're building at A-Alpha. Then we'll kind of take a step back and talk a little bit about the history of the company and spinning out from the University of Washington. And then we'll close with Q&A. But please don't hesitate to interrupt with questions as we go. So yes, there will be time at the end, but happy to also take questions live, as we go through slides. Perfect. So with that, let's jump right in. So at A-Alpha, we are building a high resolution model of biology. And we're doing that by measuring protein interactions in abundance. So looking at protein interactions on the order of millions at a time. And as I previously mentioned, many really high value therapeutics function through protein interactions. And the two main focus areas at A-Alpha are biologics and molecular glues. Both of these therapeutic areas have really significant technical challenges. So in the biologic space, when discovering and optimizing an appropriate therapeutic antibody, we need to, first of all, move as fast as we possibly can, particularly in a context of responding to an evolving infectious disease, for example. So we've done a lot of work in the COVID space and other infectious disease spaces. We want to be able to find that antibody as quickly as we possibly can. And everything in the lab just takes a lot of time. We need to be considering the affinity, so the strength of binding to a target, cross-reactivity. So for a virus, that might be binding to as many different variants of that virus as possible. For a human target, that might be binding to the human version, but also animal versions, so that the drug can be effectively tested in preclinical animal models. And then, of course, also developability. So we can't just have a really strong binder that doesn't express or that isn't thermal stable, because that's just not going to be an effective drug. Challenges also in the molecular glue space, but we'll really focus on biologics for today. At A-Alpha, we both have internal programs where we're discovering our own drugs. And we also partner and really kind of see those two things in concert, where the internal programs, we can focus on kind of the things that we learn from our partners are the perfect applications of our platform, while we continue to expand and explore new opportunities for new applications of our platform with potential partners. So in the biologic space, our internal work is a pipeline of cis-targeting immunocytokines. So these are drugs that consist of an antibody that's fused to a cytokine, where we are discovering and optimizing the antibody and also engineering that cytokine for stability, for detuning potency so that it doesn't signal systemically, and then advancing those programs internally at A-Alpha. We also have partners in the biologic space. Two that I'll mention are Gilead, where we're discovering and optimizing a therapy for HIV, and Lawrence Livermore National Lab, where we recently received an additional big chunk of funding, where the goal is rapid response to potential biothreats that may emerge sort of post-COVID. Molecular glues, similar strategy. So I think it is not at all a controversial statement anymore that machine learning is revolutionizing the way that we think about biology, science broadly, and protein science is a real hotbed for machine learning. So when we look at DeepMind's AlphaFold, when we look at the work from David Baker's lab with RosettaFold, this is a really fantastic application that gets cited across every industry of the power of machine learning. The challenge, though, is that in order for machine learning models to be really effective, they need to be fueled with really high quality, abundant data, or if not high quality, at least interoperable, unbiased, large quantity data. And that is absolutely available when we think about protein structure, because there is a really powerful database in the PDB, the protein data bank, which has essentially compiled structures of many proteins, and is highly interoperable, right? Those are very high resolution data that can be used to train those models. But as we move to protein function, the availability of that type of abundant, high quality data is really lacking. And as a result, even though this is where the real therapeutic value lies, right? Being able to design a binder that's able to neutralize a virus or that's able to kill a cancer cell, we need to understand the function of those proteins. But because the data is not available, this is a place where machine learning has been much slower to sweep in and have a really large impact. So at A-Alpha, what we are doing is using synthetic biology to measure protein-protein interactions at a massive scale, and then taking all of those data and applying machine learning to discover better drugs faster. So essentially filling that data gap using a synthetic biology platform, and then enabling those machine learning applications. And we're doing this with two core platforms. And we call these platforms AlphaSeq and AlphaBind. So AlphaSeq is that platform that Todd was alluding to, where we're essentially hijacking yeast mating as a tool for really high throughput measurements of interactions between proteins. And then we take all of those data and we train predictive models. But let's start with AlphaSeq and talk about that in a little bit more depth here. So when we perform an AlphaSeq assay, we begin with the construction of two yeast surface display libraries. So here we're expressing proteins on the surface of yeast cells. In this case, one of those libraries is a library of antibodies, and the other is a library of antigens. For different applications, these can be different types of proteins, but many of our AlphaSeq assays at A-Alpha are a library of antibodies and a library of antigens. Once we built those two libraries, we mix them in a liquid culture. And as the yeast cells are swirling around and colliding with each other in that liquid culture, they're essentially sampling protein interactions, right? If there is an antibody-antigen interaction that's sufficiently strong, then when two cells collide with each other, it will cause them to stick, almost like Velcro. And then through yeast mating, this A mating type or A sex and alpha sex cell will fuse and form a diploid cell. And through that cell fusion, the two cells combine their genetic information into that diploid cell. So now we can use sequencing, we can use next generation sequencing to sequence the DNA of this diploid cell to determine what two proteins interacted in order to form that particular cell fusion. So instead of having your wild type yeast strains that express their own native proteins on their surface that cause them to stick to each other, the way that we engineered the system is we essentially shaved those proteins off so that the cells are no longer able to stick. And then we express these libraries of proteins that we're interested in to see if they can recapitulate that sticking, which has this next generation sequencing readout. What's important about this process is that it's affinity driven. So strong protein interactions result in many cellular fusions, weaker protein interactions result in fewer cellular fusions. And so when we're performing next-gen sequencing, what we're doing is we're counting the number of cell fusions that occur from every possible pairwise protein interaction. And that allows us to generate a quantitative affinity score essentially for every one of those pairwise protein interactions. So the output from an AlphaSeq assay looks like this heat map in the middle here. In this example, we're measuring a total of 3 million protein-protein interactions. In this example, that's 15,000 antibodies and 200 targets. This example is in the infectious disease space. So our goal is to identify and optimize antibodies that are binding as cross-reactively as possible with these 200 targets, which is a problem that's otherwise really, really difficult to approach when you can only measure antibodies binding to a single target at a time. So this type of multidimensionality is really important for solving those types of problems. In this heat map, dark blue indicates strong binding, light blue indicates weak binding. But even at this scale, we're getting really, really strong quantitative information. So if we take a subset from these 3 million interactions and actually use a low throughput technology where we express those proteins, purify them, and measure their affinities one at a time, we see a very, very strong quantitative correlation between AlphaSeq and this low throughput approach, which is called biolayer interferometry. So essentially, we're able to get the same quantitative information from a low throughput assay, where we're measuring one interaction at a time, but here we're measuring 3 million at a time. Once we've generated this type of a data set, sometimes we'll have identified a sequence that shows appropriate cross-reactivity, for example, and now we're done, right? We're ready to move that antibody forward. More often, we need to go through multiple rounds of experimentation in order to optimize an antibody for the plethora of a unique characteristics that we're trying to optimize for. So in that case, we'll take all of these data, we'll use them to fine tune our AlphaBind model, which will then predict new antibody sequences that are expected to have improved properties. We'll synthesize those new antibodies and test them in a second iteration of an AlphaSeq assay and proceed as needed until we found the appropriate therapeutic. Just to maybe make this a little bit more real and kind of look at some smaller scale data here. Here is data that is looking at interactions between antibodies against SARS-CoV-2, so against the coronavirus, as well as other related coronaviruses. So one of the real challenges within any infectious disease space, but certainly true for the coronavirus pandemic, is that because the virus evolves very rapidly, it's not at all obvious what therapeutic antibody is going to work against any given variant. And ideally, we would have an antibody that's able to bind to as many of those variants as possible, so that we don't need to constantly reinvent a new drug every time the virus mutates. So in this case, what we're looking at in this inlay from an AlphaSeq assay. So again, in the single AlphaSeq assay, we're generating lots and lots of data. This is a very small subset of that data. Here we're looking at a panel of anti-CoV antibodies, and we're looking at how they bind to multiple replicates of the wild type Wuhan strain of the coronavirus, other related coronaviruses, so including SARS-CoV-1 and others, and then different mutational variants and circulating variants of the coronavirus. And what we can see is that, for some of them, they maintain binding to quite a few of these. So, this is a pretty good candidate that's showing pretty broad cross-reactivity to all of these different variants. But many of the therapeutic antibodies really show pretty poor binding to some of these circulating variants. And so with data like this, we're able to both identify those antibodies that are going to be appropriate against a particular strain, and also start to make predictions about how we can optimize an antibody so that it's going to be more applicable for potential future circulating strains. So kind of one quite visceral example of how we can apply this platform. Great. So when we think about then expanding this into machine learning applications, I've already mentioned some of this, but when we look at the available data in either the public domain, kind of within academia, within other companies, there's a real problem where protein binding data sets are typically quite small. They're typically not interoperable. So if you are measuring protein interactions with different instruments or even different protein concentrations or buffer conditions, often those data are not readily comparable to each other. And then many approaches for measuring protein interactions are really just binary. So we're determining, is there a protein interaction or not, but not getting a quantitative readout for the affinity of that interaction. And so, our solution is, generating data with a platform like AlphaSeq allows us to generate an abundance of quantitative data that's all interoperable. And then we can use those data to train effective machine learning models. So there are a lot of attributes of AlphaSeq data that make these data sets particularly well suited for training models. For example, the data is library on library. So we're not just looking at how a large set of antibodies bind to a single target, but rather we're looking how a large set of antibodies bind to a large diversity of targets. And particularly if we're trying to ask a machine learning model to predict which antibody or what novel antibody sequence is going to bind to a novel target that we've never seen before, we better have seen a large diversity of targets so that the model can start to learn how the mutations in that novel target that we haven't seen before relate to all the other targets that the model has seen before. So that library on library two-dimensionality is really, really important for these applications. Speed is also really important, right? We want to be able to iterate through cycles of experimentation and computation really fast. And because we're working with yeast, which is a simple single cellular model organism, we can get data from DNA in-house in about a month. The platform is versatile, so it's compatible with lots of different types of protein interactions. Antibody antigen being one core one that we're spending a lot of time and investment in, and then also the data that we generate is quantitative, so not just does an interaction exist or not, but what is the strength of that interaction? Just to kind of give a sense for the scale of lack of data availability broadly, the largest public repository of protein interaction measurements is a database called BioGrid. And right now, this contains about 1.4 million literature-curated protein-protein interaction measurements. As I mentioned in a previous slide, in a single assay, we're able to measure about 3 million protein interactions, and we now have a database that contains over 1 billion. So, again, the publicly available data around protein function, around protein binding, is far, far too small to train sophisticated machine learning models. So what are we then kind of using this for? How are we moving this forward? So there are kind of two key places where we're using experimentation and computation. One of them is to really improve our methods, right? To learn how we can discover and optimize antibodies better and faster by using high throughput experimentation as a tool for rapid hypothesis testing. And then the second that we'll get to soon is taking those best models and applying them for the discovery and optimization of new antibodies. So one of the key problems in antibody discovery and antibody development is once we've identified a starting point antibody, how do we make it better? How do we optimize it? And again, there are a lot of variables for which we're optimizing. So we're optimizing for affinity to its target, potentially cross-reactivity to lots of different targets, specificity. We don't want it to bind to anything that's going to cause off-target toxicity, developability properties. So we're talking about a really complex, multi-dimensional optimization problem. The challenge here is that with combinatorial diversity, the potential sequence space, even starting from a parental antibody, very, very quickly explodes. So if we start with a parental antibody and we want to look at just single amino acid mutations within CDRs, we're looking at, on the order of hundreds of single point mutants. When we combine to double mutants, we're talking about hundreds of thousands, triple mutants, hundreds of millions. We might want to look at an edit distance of 10, 15, 20 amino acids away from parental. At that point, the diversity is far, far too large to ever capture experimentally. So the old school approach has been identifying favorable mutations one at a time. So looking at just the single mutations and then trying to stack them, right? Trying to figure out the ones that seem the best and combine them manually to test a relatively small number of those combinations. Machine learning can do much, much better. And there's we have had lots of success across the industry of machine learning being able to solve this problem much better than just this iterative stacking. But there are lots of different strategies that you might employ for using machine learning to solve this problem. So how do we do this the best? How do we optimize our optimization? What we're able to do with the volume of data that we're able to generate at A-Alphabio is do parallel perspective model validation. So essentially try lots of different approaches, have each of them predict lots of antibodies, and then go back to the lab and test all of them. So in this case, we're looking at two key dimensions to how we're thinking about these models. One of them is model complexity, and the other is the quantity of pre-training data that we're using for that model. We can then build a number of different model architectures, have each of those make predictions of antibodies that are expected to have improved properties, and then test them head to head. So just to kind of look at a small scale example of that, here we're combining antibody-specific data from a single AlphaSeq assay with unrelated antibody binding data from our database of 1 billion protein interactions. We're using different amounts of this pre-training data, as well as different model architectures to train 4 distinct models. Each of those models are now being asked to make predictions of sequences that are different number of step sizes away from a parental antibody. So 2 mutations away, 3 mutations away, 4 mutations away, et cetera. Each one is predicting 750 unique antibodies that are at each of those edit distances. So each one of these models is making a total of 7,500 predictions. We're synthesizing all of those antibodies, putting them into an AlphaSeq assay, and actually experimentally testing which of these model architectures is performing the best. And what we can see with model one is already industry competitive. So, being able to go straight from this starting point data to being able to predict antibodies that are better than that parental, that are 6 mutations away or 5 mutations away, is pretty much the state of the art for the field. But by being able to test lots of different approaches and using all of our pre-training data, you can see that our current kind of production AlphaBind model can do much, much better than that. So we can go out to 11 or actually even considerably further edit distance away from the parental antibody and still have really, really good binding. So that's how we can essentially use AlphaSeq and AlphaBind as a tool for improving our methods for training models and identifying that optimal model for antibody optimization. Next, we want to essentially take those learnings and apply them for the discovery and optimization of a new antibody. And for that, at A-Alpha, we have a pretty standardized workflow where we are starting with some goal in mind. We have an in-house phage display library. So we have a library of antibodies that's displayed on the surface of phage that we use for panning. We then move into phage panning. We then go through multiple iterations of AlphaSeq and AlphaBind to find an optimized antibody that meets some specific therapeutic criteria. And we'll kind of go through some of these steps one at a time to talk about exactly how we do that. So we'll start with this phage library. We want to have a massively diverse library of phage that we can use to discover novel antibodies against any target that we're interested in binding to. So this is a completely naive library, so not biased towards binding to any particular target. But we want as many of those antibodies in that library to be highly developable as possible, right? So any antibody that we pick out, we want to make sure that it might be a suitable drug. And for that, we want it to be as close to a human antibody as possible. So we actually start with our machine learning at this step, where we're designing this antibody library, this phage library, by looking at millions and millions of native human antibody sequences, training a machine learning model to learn what does a human antibody look like. So how do we identify potential liabilities that might make an antibody not suitable for production or might cause immunogenicity or some other problem? And then we can synthesize that distinct library where these are antibodies that are not human. They've never been sequenced out of a human sample, but computationally, they're indistinguishable from human. They have all of the human properties. Once we have that library, we can proceed with antibody discovery. So for that, we begin with phage panning. So with phage panning, the advantage of using phage over a technology like yeast is that we can move to a really, really massive diversity. So we have a phage library that contains about 10 to the 11th unique antibodies, which is a really, really big sequence space. And when we're doing phage panning, we're approaching this a little bit differently than most companies that do phage panning for antibody discovery. What most companies will do is they'll start with that really massive diversity, and they'll pan down until they have a pretty small number of antibodies that are really strong binders. So maybe they have a dozen or a couple of dozen strong binders to their target. We don't want to go that far. So we do what we call low stringency phage panning. So we typically only do 2 rounds, such that we still maintain an antibody diversity of about 10 to the 5th. At that point, we can take all of those antibodies, so about 10 to the 5th antibodies, and clone them into AlphaSeq so that we can identify from that large diversity all of those antibodies that exhibit the properties that we're looking for in a potential therapeutic. So here's an example where we've done a phage panning against human TIGIT, which is a high value target for us and many others. It's seen some clinical challenges, but it's still a potentially a really exciting target. We did 2 rounds of panning, subcloned into AlphaSeq, and now we're characterizing all of those TIGIT binders for binding to human TIGIT, We're looking at cross-reactivity to mouse TIGIT, which isn't something that we panned for, but we were hoping to see this in some small fraction of binders that we identified. And then we're also looking for non-specific binding against closely related proteins and completely unrelated proteins. So we want to make sure that our antibodies are highly specific for their target. And we were able to, fortunately, identify a number of antibodies that were quite potent and also showed this cross-reactive property of binding both to human and to mouse forms of TIGIT. So from there, we can move to iterations of AlphaSeq and AlphaBind to optimize those antibodies for the properties that we're most interested in, namely developability and affinity both to human and to mouse TIGIT. And what we can see here is that, as we move away in mutational space, away from our parental, we are able to see that by a first iteration, so we do an iteration 0, which is just single point mutations, and then we do an iteration 1, which is where we're starting to combine those mutations using our AlphaBind model. And by that iteration 1, we're able to identify, in this case, antibodies many mutations out that are not only preserving binding for human or strengthening binding to human, but that are also maintaining that cross-reactivity to mouse. So this is a more complex design challenge, which is why in this case we're not able to go out to say 15 mutations because we're optimizing both for on-target binding to human TIGIT and also cross-reactive binding to mouse TIGIT. When we actually look at the properties of these antibodies, so our parental antibody expressed well and had a pretty good affinity to humans, so a little bit subnanomolar, and a pretty weak binding to mouse. So detectable, not terrible, but not nearly ready to be used as a surrogate for mouse binding. So after this single iteration 1 data set, in this case, we were able to identify 2 distinct variants from this optimization campaign, one with 3 mutations and one with 4 that are still showing good expression, now have very, very strong human binding, so no detectable off rate by biointerferometry, and now we're down to single-digit nanomolar binders against a mouse TIGIT. So from one round of optimization, we're able to achieve these really, really nice binders that can be moved forward into our subsequent applications. So one other kind of quick application that I want to touch on before we close here is how we can also use AlphaSeq data and our AlphaBind models to improve how we predict the structure of protein-protein complexes, which is a really big challenge, especially for predicting interactions between antibodies and antigens. So when we use a technology like AlphaFold Multimer, which hopefully some folks are familiar with and have used for predicting how proteins come together in structural space and bind to each other, what this technology does is, starting from protein 1 and protein 2, it performs a query to do a multiple sequence alignment and identify different animal homologs of the proteins that we've queried. It looks in coevolution space for different amino acid positions across those proteins that co-evolve, and essentially learns that those positions that co-evolve are very likely immediately adjacent at the protein interface. So it can use that coevolution information through this MSA embedding to make predictions of how those proteins come together in space, recognizing that those co-evolved amino acids are likely in close proximity across that protein-protein interface. The problem with antibodies and antigens is that they don't co-evolve in the same way that kinase and its substrate co-evolve, right? An antibody is essentially evolving on its own, kind of live to a particular pathogen. It's not a coevolution across species. And so this type of information is not possible to get from any kind of database. And without this information, technologies like AlphaFold Multimer really struggle to be able to perform these types of structure predictions. So what we're able to do with AlphaSeq is essentially generate these types of data synthetically. So we can make mutations in both of our proteins, so make mutations in our antibody and make mutations in our antigen. In this case, we're making all possible single mutations. So at every position, we're mutating to all 19 alternative amino acids. And then with those two libraries, we can perform an AlphaSeq assay to essentially learn how all of those single point mutations relate to each other. And what we're looking for is something called a synergistic mutation. So essentially the idea, if we have an antibody and an antigen, these two proteins might interact, right, on their own, and hopefully they do, because that's our starting point. It's possible that if we make a mutation in our antibody, we might completely eliminate binding. It's also possible that if we make a particular mutation in our antigen, we might completely eliminate binding. But if we stack these two mutations together, so now we're looking at that mutant in the antibody that eliminated binding and the mutant in the antigen that eliminated binding, it's possible that those two mutations essentially cancel each other out, such that the two mutants are able to bind to each other. That's called a synergistic mutation. And it essentially indicates that those two amino acid positions are probably right next to each other at the protein interface. So this is essentially the same information that could otherwise be gathered from coevolution. We're now generating it synthetically, plugging that into an MSA embedding, and now generating much higher quality, higher confidence predictions of how antibodies and antigens are able to bind to each other. And just to kind of show some data to that effect, looking at the prediction of an antibody and antigen binding to each other, AlphaFold confidence for those two proteins when given sequence alone is pretty darn poor. It's kind of all over the place. When we inject AlphaSeq data, we're able to really dramatically improve that confidence. And when we have looked at kind of comparisons between predictions and known crystal structures for particular antibody antigen interactions, we can confirm that we get much higher resolution, higher confidence predictions. Great. So with that, kind of taking a step back. So again, at A-Alpha, we are working on things internally. We're also working on things with our partners. So in the biologic space, again, our internal work is focused on precision engineered biologics, where we're engineering antibodies fused to cytokines, such that they selectively signal particular cells without activating off-target cells. And then in the molecular glue space, we're using many of the same approaches for understanding how E3 ligases and potential neosubstrates come together in space so that we can rationally discover small molecules that can enhance those particular interactions. So again, at A-Alpha, we are harnessing synthetic biology and machine learning to measure, predict, and engineer protein-protein interactions. So this is measuring binding in abundance so that we can really understand biology, predicting how proteins interact so that we can move faster and more reliably, and engineering precision binding so that we can do things that otherwise just wouldn't be possible with traditional experimental approaches. So with that, I know we don't have a lot of time here, but want to touch quickly on a brief history of A-Alpha. So we spun out officially in 2018, but really started building the foundations for A-Alpha in 2017. So not that long ago, we were a team of 2 full-time employees. We were funded by grants and awards. We were very much in a proof of concept stage. Had some ideas for the right applications of our platform, but there was a lot of fuzziness. We relied very heavily on a lot of resources that were provided to us through the University of Washington. Todd mentioned the CoMotion Gap Fund, which was a postdoc funding for me to really devote a full year to thinking about how to commercialize this technology, among many others. And then funding started with University of Washington awards, for example, at the business plan competition, a first grant from the NSF, so a small business innovation research grant, some funding from the Gates Foundation, and then so far we've raised 3 rounds of funding, so a seed, and then a Series A and a Series A2, totaling about $50 million, and then additional partnership revenue and funding from the DoD and other government agencies on top of that. So today we are a team of 52. Our funding is both from venture and also revenue. And our stage is very much, building an internal pipeline of drugs within A-Alpha and also partnering with some of the leading pharmaceutical companies. Um, kind of thinking back to the end of my PhD and starting to kind of get the company off the ground, kind of what were we doing? What was I doing during this time? So during the final year of my PhD, I was thinking a lot about graduating. As Todd mentioned, it is really valuable in this space to have an advanced degree. But also thinking about intellectual property, which is really, really important for getting a biotech company off the ground. So working with University of Washington CoMotion to get our intellectual property kind of all in order. So filing a provisional patent. And then also really doing a lot of work to evaluate the opportunity. So going out and talking with people at pharmaceutical companies and in biotechs to understand whether the technology that we were building really had a place to and did it really make sense to start a company around it. During that postdoc year, this was where I spent a lot of my time on what's called customer discovery. So getting out and talking with folks in pharma, really evaluating what are the right applications of the platform. Also, as a part of this, doing a lot of learning and a lot of networking. So through business plan competitions, through introductions to venture capital and also pharma companies. This time was also fantastic for applying for grants. So because we had funding for a year, we knew that had a pretty sharp timeline cutoff. And so we wanted to make sure that we were prepared with funding, when that postdoc year ended. So both talking with venture capital if we needed to go that route from the very early days, but we were really hoping that we could bring some non-dilutive capital in first from a grant, and that's fortunately what we were able to do with that first National Science Foundation grant. And then in the first year after spinning out, this is where we were really starting to think about building the foundations of the company. So going from 2 people to 5 people and then to 14 people over the course of more than just 1 year, but starting with turning those venture capital relationships into our first round of funding. Converting some of our BD relationships into early pilots and partnerships with pharma companies, and then starting to build the team. So with that, I want to thank everyone again so much for your attention and learning more about what we're doing at A-Alpha. And I'd be very, very happy to take any questions that anyone has.
Thank you so much, David. I think it's a really tour de force and come a long way since Genome Startup Day. I have to say. So
go ahead, Sandy.
I was wondering if I could ask the first question.
Sure.
Okay, since this is my idea. No, you've talked a lot about the amazing computational work that your company is doing, but you've also mentioned that there were screening assays as well. And with 50 people, I'm really interested in knowing what kinds of lab skills are you looking for when you hire entry-level technicians in your company?
Yeah, great question. So really depends very much on the role. So we have what we call a technical operations and engineering team, which is the team that is essentially running AlphaSeq on a daily basis, a week in, week out basis. So they are getting DNA in the door from a DNA vendor, building yeast libraries, transforming them, characterizing those libraries, doing AlphaSeq assays, taking genome extractions from those cells, running them through next-gen sequencing. So these are roles that really require good hands in the lab, right? Just really good kind of core microbiology skills. Obviously, no one else is doing AlphaSeq assays, so the expectation is not that anyone is familiar with our assay, having done it in a lab. It's really about really good techniques, doing PCRs, doing cell cultures. Any experience with next-gen sequencing, we use Nanopore, we use PacBio, we use Illumina sequencing. So any NGS experience is really, really valuable. So that's within our technical operations team. We also have other teams that spend a lot of their time in the lab, for example, characterizing protein once it's expressed, or expressing and purifying protein and doing kind of core characterizations on those proteins, and then testing those proteins both in cell-based assays and in animals. So there are lots of kind of different specialized sets of expertise in the lab as well. About half, a little over half of our company spends a lot of time in the lab. So those skills are really, really paramount.
Thanks. Okay, now we'll go with hands raised. So Keith.
Thank you. David, can you hear me?
Yes. Hi, Keith.
Excellent presentation. You're doing some amazing work there. The big question that comes to my mind is the final product you're selling, what is that and what's the cost? So to elaborate on that, a company comes to you and says, hey, I got this Mpox and I need to find a suitable vaccine. And do you just run with that, or do they have to gear more towards specifics?
Yeah, great question. So again, we are both doing work with partners and we're working on our internal pipeline. So the answer is quite different for those two spaces. So with partners, an example of a problem that a partner might approach us with. So for example, it's kind of rooted in something real world. Gilead is the leader in HIV therapies. They do a really, really good job in HIV. They have a wonderful virology team. One of the big challenges in developing a therapeutic against HIV is you often need to make them specifically against kind of different subsets of the virus. It's very difficult to find something that's pan-virus, and those therapies often get evaded, right, as the virus continues to mutate. They have not been able to generate the diversity of binding data that we are able to with AlphaSeq. So they essentially came to us and said, we have some starting points for potential biologics that could, neutralize some subsets of HIV. We want you to help us find one that's far more cross-reactive. So we work with their scientists to kind of better understand the problem, to understand the disease biology so that we're making sure to design our experiments appropriately. And then we essentially take that in-house, right? We build those variants of potential therapeutics. We build large libraries consisting of massive viral diversity. We generate those data sets to look for cross-reactivity. We periodically report back to the Gilead team and have collaborative discussions about, well, okay, we're able to bind to this family really, really well. This one's more challenging. How do we consider these trade-offs? We kind of make all of those decisions together and ultimately deliver a panel of optimized therapies back to Gilead for them to test in-house to essentially validate that they're able to effectively neutralize against that broad cross-reactivity of virus. And from there, Gilead runs with it. So they can proceed as they wish through preclinical, through IND-enabling studies, ultimately into the clinic, and then they would market that drug. That is different from our internal program. So we're mostly oncology on cancer therapies at A-Alpha internally. There, we're in kind of the stage of discovering molecules, testing those in animals, and proceeding towards IND. But we're still a long, long way from generating revenue from molecules that we've discovered in-house.
How do you set up the cost structure with the first methodology? Is it
Yeah,
a blank check or a program specific limitations?
So our partnerships are structured with a number of different kind of payouts along the way. So a partner will pay us upfront some lump sum, which is essentially access to our technology and a recognition that they're going to be taking a bunch of our bandwidth. So there's an upfront payment. There are then research payments as we complete different deliverables. We get additional kind of checks along the way. And then once we've delivered those molecules, they have the right to take a license, which requires them to make a payment. We're now done, right? So we have no more costs associated with the program, but we do have a risk share. So essentially, as that molecule now gets into the clinic and proceeds through clinical trials, when milestones are hit, the partner pays us pre-negotiated fees for hitting those milestones. And then when that molecule is on the market, we get a small sliver of any revenue that gets made in the form of royalty payments. So it's a potentially kind of long road, but that starts with an upfront payment at the very beginning.
Thank you. I do have other questions, but I'm going to let other people have a chance to sit with you.
Right, and we're aware of your next talk at 9. So, Leah.
Yeah, great talk. Thank you. I've been interested in your company for a while, so this is really, really a special treat. Um, so my question is actually around your funding from the Gates Foundation and some of the rationale there in terms of infectious disease, small markets, forgotten markets. Well, not forgotten, but sometimes undervalued.
Yep.
And like, what is your strategy for helping to enable some molecules that might be very applicable to infectious disease to go forward? And also, if you have examples of, indications that might be of particular interest, that would be great.
Yeah, absolutely. So I think, it's no coincidence that a lot of our partnerships in the biologic space are infectious disease focused. It's a great place for us to have impact and to use our technology for an application that is perfectly suited for our capabilities, right? Being able to measure binding against a really large diversity of antigens, and optimize for cross-reactivity. I mean, that is a perfect application. As you mentioned, the market is tough, right? And so that is not where we are focused with our internal programs, which is perfect, right? So we can essentially work with partners non-competitively, right? We don't have to worry about kind of staking a claim to those targets. And that's why, with our work with Gilead, we're working, on HIV. Our work with the Gates Foundation actually started pre-COVID. We were working with E. coli and Campylobacter. And then when the pandemic started, we added COVID to the mix. With our work with Lawrence Livermore National Lab, that's DoD funded. This is a biothreat preparedness project. So we're working, we started with COVID, but now we've expanded to different, really kind of high value infectious diseases that could represent future biothreats, either naturally evolved or weaponized. So we are heavily invested through partnerships in a number of different applications in the infectious disease space, which again, I think is a wonderful way to kind of have impact with those applications, while also recognizing that our investors are not going to want to put lots of money in A-Alpha for us to develop those therapies ourselves.
Thanks, David. So we have time for one more question, and Aaron, I believe you're it.
Yeah, thank you. Hi, David. Thank you. Very, very well done. A great presentation. I'm Aaron. So I used to do some protein engineering before and did some basic sites, the directed evolution to build the biosensor for some pharmacologic agent. So, but now I'm in the education and workforce development space, and also one of the team leaders for this hackathon. Our group trying to build classroom activity that's affordable to be done at a community college or high school classroom to teach students about protein engineering. Now, one thing that, sort of like the look ahead, we were thinking about sort of like combining exactly what you mentioned here, using the machine learning to predict certain antigen antibody interaction. However, like you mentioned, the public database is not large enough to predict that fundamental biochemistry in amino acid interaction. Not to mention there's complication with synergistic combination as well. So what would you sort of like, do you have any advice for someone like me who developed this kind of classroom activity, knowing that a lot of these protein-protein interaction database are not accessible because they are owned by like private company, like the big Amgen of the world and things like that. So
Yeah. So I think that, for an education application, there are some data sets that have been made public that can be kind of really, really nice kind of places to start to dig into what these types of high throughput data look like and how you can use them to train machine learning models. I think what is not possible using public data, and to be frank, not yet possible using even A-alpha data, is to move towards increasingly generalizable models, right? So the dream is you give me the sequence of any antigen, and I will be able to predict 1,000 antibodies that bind to, precisely defined epitopes across that antigen. That is not possible, right? We hope to get there, but that is not possible. There is still not enough data to enable those types of applications. But there are specific data sets around antibodies binding to a single epitope, for example. So the data, for example, that I showed a number of slides ago with the SARS-CoV-2, so looking at different antibodies binding to different variants of SARS-CoV-2, that is a figure from a paper that we published in 2022 that essentially contained 1 or 2 alpha-seq assays worth of protein-protein interaction data as part of that publication. So that's a place where you can go and you can use that data. You can also look at lots of other potential publications of it. But there are these types of data sets that have been made available, not nearly enough to enable kind of the broad generalizable problem to be solved, but that can absolutely be used to train specific models for specific problems that teach the same skills that will ultimately be needed for those larger scale models.
Thank you.
Well, thank you, David. Once again, everybody, do your virtual clapping. And we really appreciate you joining us today. And good luck with your next talk.
Thank you very much. Yeah, and really, thanks so much to everyone for your attention and great questions. Yeah, I mean, it just sounds like a really wonderful experience that you all are going through here. Yeah, if there's any way that I can be helpful in the future, please don't hesitate to reach out. Yeah, Sandra and Todd can please feel free to share my email. So thanks, everyone.
We were hoping you would ask. And thanks again.
Yep.
Thanks.
Yep. All right.
Okay.